Okay, we are at the top of the hour, so we'll get started. I am Courtney Cheetah, and I'm a program associate with Wild Farm Alliance. Thanks for joining us today for the Whole Farm Pest Sustainable Pest Management course. This is the final lesson of 12. Our mission here at Wild Farm Alliance is to promote a healthy, vibrant agriculture that protects and restores wild nature, and we help bring wild nature back to farms. Just as a reminder that all participants are muted. If you're experiencing any technical issues, there are moderators in the chat to help. And please use the chat to ask questions for the presenters. Our 2050 vision is to see 1 million nest boxes and perches on farms, hedgerows of habitat to the length of the moon and back, and to help support all the different kinds of beneficials on the farm, restoring the riparian areas because they're the lifeblood of the landscape. And we really see avian pest control as part of the toolbox for managing crops. Next, I want to share a new resource that we're building and invite you to join us. If you are an expert in sustainable pest management and would like to showcase your expertise to growers and potential clients, you can just do that by completing the profile builder. Your location will then be featured on the interactive map with detailed pop-up information, enabling growers to quickly find experts specializing in their crops, addressing their unique needs, and located in their areas. And what's awesome is this is up and live. So you can go check out the directory right now. If you're looking for a sustainable pest management expert, a pest control advisor, or a certified crop advisor, you can now find an expert in your area using the SPM directory. And you can find the link for that in the chat as well. Today's webinar is Sustainable Pest Management Encompasses Whole Farm Systems Thinking with David Hedrick from Cal Poly SOL and Pest Control Advisor Devin Carroll. They'll be up in just a second. We have on our website, we have under the events 2 different courses that are free that have continuing education credit available for them about supporting avian pest control. Participants will learn about research that shows the benefits of installing nest boxes and habitat in and around farms to attract beneficial birds that promote valuable pest control services. And you can take those on demand at your own pace. And the courses are available through the end of December of this year, 2025. We are planning more events related to sustainable pest management, so keep an eye out for those. We're not sure how quick they're going to be happening, but they'll be happening sometime later this year. And then please consider attending all the rest of the lessons in this series. If you've missed any of the earlier ones, they've been really great. The recordings are up on our website under the online courses under news and events on our website, wildfarmalliance.org. We have continuing credits education available from DPR and ASA. And if you're seeking any of those credits, we will have a pop-up poll after each speaker that we'd like you to participate in. And then at the end, there will be a link to a quiz and evaluation form where you can share your license number with us for that credit. Next, I'm going to pass this off to David. If you have any questions, please put them in the chat, or if you're having any issues, let us know and we can try to help. And I will stop sharing and let David share his screen and we can go from there.
All right. Well, good morning, all you folks out in Zoom land. So this morning, what I want to talk about is the use of the California red scale sex pheromone in order to help control California red scale in commercial citrus production. Now, my graduate student, Joel Leonard, he and I, working with Beth Grafton Cardwell, who was the citrus IPM specialist at the time, this took place in 2018. We wanted to look at pheromones and the incentive for using and trying to use pheromones to control California red scale comes from the fact that there's so many factors out there affecting insecticide use for California red scale. One of them is increased trade regulations, like the use of the impact of MRLs, the maximum residue limits. What's happening there is, of course, for export, that's dictating what kinds of pesticides can be used, how much can be used, when they can be used. And so that is kind of influencing a drop down in insecticide use. There's also the issue of quarantine regulations for invasive species like ACP. In that case, there's an increase in insecticide use in order to clean up the citrus to move it for packing and distribution and so forth. If that's the case and you're adding more insecticides in, then that's going to make it so that growers are not going to want to purchase aphitis wasps for augmentative biological control of California red scale. And so there's going to be a reduction in the use of natural enemies and an increase in the use of insecticides. We also have the issue of changing weather patterns. With warmer winters that we've been having, we're experiencing more generations of California red scale. In fact, we're up to five, whereas it used to be three to four. With five generations, you've got more population, increased numbers, which then leads to more insecticide applications. And then we've got the granddaddy of them all, and that is insecticide resistance. So we've got kind of a catch-22. We've got factors that are forcing growers to reduce the number of insecticides, but then we've got factors that are forcing growers to increase the number of insecticides. And if we go with the increase in insecticide use, well, then we're already in a problem because of the current amount of insecticide resistance that's already documented. We have documented cases to most of the major chemical classes with California red scale. And you can see in this map on the right, this is the major growing area for citrus in California from Bakersfield up to Sanger. And if there's a yellow dot, those are populations that have low resistance to these major chemical classes, all the way up to the red dots where there's high resistance. So pretty much all through that growing area, you've got some resistance, some significant resistance to the major chemical classes of insecticides. There are some areas where there is still some susceptibility in green. And there's still some products like buprofuscin, spirotetramat, and narrow range oils where we've got some susceptibility still in those populations. But what's going to happen is if you start using those and abusing those, then we're going to have resistance to that. So the insecticide issue is just presenting a lot of problems. And so that's the impetus to look for another kind of a tool, and the use of pheromones is one such tool that can reduce the populations and also play well with things like biological control. I need to go over a little bit with the life cycle here, so bear with me. In order to be able to tell this story and have an understanding of what the pheromone is doing, I need to go over this just a little bit. So on the left, we have a reproducing female, meaning that she's laying eggs. And those eggs hatch out, and the crawler emerges from underneath that female. And if it's a male crawler, what's going to happen is it's going to molt and become this second instar male that's then going to go through its pupal stages to eventually become a mobile adult with wings, legs, and so forth, so it can fly, it can walk. Now, if that crawler is a female, what's going to happen is that it's going to go through a couple of molts to become a third instar female. And a third instar female is the one that is going to be receptive to mating. So she's the one that's producing a pheromone. Okay. And that's where the mating takes place. So that male is going to go find that third instar female that's producing a pheromone. Now, what happens is that it's similar to a lepidopteran system that we might be all familiar with, and males flying to find females. Okay, so they follow the pheromone to find a female to mate with. But because we have a plant surface that may be covered with lots and lots of scales, you might have a male scale emerge and literally be standing right next to a receptive third instar female. And so they can just turn around and mate. So we've got a little bit of an issue here about, yeah, the flying and maybe disrupting the males with the pheromone, but then we have this proximity issue. So I'll get to that in a second. Once mated, this female scale is going to become encased in wax underneath that scale cover. And what's going to happen is that's going to make it difficult for her to be contacted by insecticides and also for parasitoids to attack her. Speaking of parasitoids, that Aphytis wasp that growers use for augmentative biological control prefers that unmated third instar female. So that's what they're looking for. And that's going to come up here in a second. Okay, now on to the experimentation that we did. Basically, we took this pheromone, this synthetic pheromone that's commercially available that most growers in citrus use to monitor red scale populations. So they put the pheromone in a tab on a sticky card and hang a few out in a block, and then they go and see how many males were attracted to the sticky card. Well, for the disruption research, what we did was we took that same tab that you see there on the right and we put 180 dispensers per acre. That there's about 109 trees per acre, so we were over the number of trees. So at least one tree got one. We put them about 6 feet high inside the canopy, and then so we just permeated the entire block with this pheromone. Now, the size of the blocks that we were using were 40-acre blocks. So we wanted to be able to demonstrate that this was going to work in commercial size citrus. So the treatment blocks, the control blocks, they were all 40 acres. And so then we went back to measure how is this pheromone affecting the California red scale populations. And what we did was we put up traps like you would to monitor male scales. So we had the regular traps up and we looked at their flight data, how many males were getting attracted to these traps. Then the bottom line, we looked at how many fruit were being infested by those scales. In addition, we wanted to see if there was something going on with the mating. So what we did was we took lemons that were infested with virgin third instar females and we hung them in treated and untreated sections of these blocks and left them out for 2 weeks. Then we went and got them, retrieved them from the field, and looked to see if any of these females had mated and if there was any parasitism taking place. All right, so the results show that with the trap counts, so we went and retrieved the traps from these orchards, and we saw that we got excellent suppression of male flights all throughout the season. And in fact, when we had the pheromone disruption going with lower initial populations and larger acreage, we got 90 to 99% control of California red scale. We shut down the male flights. But the important one with the fruit infestations, what we showed was that heavily infested fruit, which is greater than 10 scale per fruit, we reduced that to less than 0.5% at the end of the season. In contrast, in untreated areas where there was no pheromone, there was an average of 5.6 heavily infested fruit. So we lowered that by an order of magnitude. Now, with the mating trial, this was interesting because less than 3% of the female California red scale mated and developed eggs in those pheromone treated areas. So what the bar graph on the right is showing you is that the red bars are where there's no pheromone. And in that case, the third instars females mated and moved on to the adult stage, whereas in the areas that had the pheromone represented by the blue bars, those females remained stuck in the third instar and they never mated and they never moved on to become an adult, a gravid female laying eggs. So that's really interesting. So what we see is that there's kind of two factors going on here. Clearly, we shut down males being able to find these traps. In other words, being able to fly and find receptive females. Shut that down, 90 to 99%. But we also shut down their ability to mate. So shutting down the male flights is very much like what we see with the Lepidopteran approach, but shutting down the mating is very different because we have males standing right next to females. And what our research demonstrated was that mating was delayed. So 97% of the females did not move on and mate and become gravid females. And that lasted for at least 2 weeks. Now, the physiological mechanism for how that superabundance of pheromone delays mating remains unknown, but it is still important because delays in mating, that reduced the female attractiveness. That led to fewer copulations, and that led to a reduction in the number of offspring, basically a crash in the population. It also resulted in production of larger female scales and an extended time in that third instar stage. And those are advantageous for parasitism by aphytis because aphytis wants that unmated female. And if it's out there for a longer period of time, that makes them more vulnerable to parasitism. But here's the kicker. A fetus will produce more females in larger scales. So if they find a large female third instar scale, they will lay a female egg in that scale. So the aphytis populations will be producing more females, therefore more scales, therefore more biological control. So this pheromone disruption, whatever's happening, does play well to biological control. So some other conclusions that we came up with is that if we deploy the dispensers before the first flight, that's more effective than waiting until after. So putting them in early when the populations are low works better. But we know that putting them in early in the season means that you may have wet soils to deal with, and they may hamper placement in the trees. We understand that. We also know that the larger area that you put the dispensers out, the better. So this is an area-wide approach. The greater the coverage, the greater the reduction in the scale populations. And an update. Currently, what we see in orchards that are using this tactic is that the use of the dispensers or the tabs has changed from every tree to every other row and then switching rows the following year because the pheromone lasts greater than a year. And that's going to cut back significantly on costs. Turns out the height in the tree doesn't matter, so you got some latitude there, but the surface area of the tree matters. So hedging and pruning to get better permeation of the pheromone is key. Now, in the past, we know that citrus pest management has been geared around California red scale control. It's the key pest. And if you were going to do a proper California red scale insecticide application, that's going to be 1,000 gallons of water per acre, and growers were going up to 3 times per year. That's a lot of resources. And right now, today, it's difficult to find California red scale in well-managed orchards because the pheromone will provide complete control. And so far, no resistance has developed to the pheromones and none likely to develop anytime soon. And I know that some growers think that insecticide resistance is fake and that degree days aren't real, but in spite of that skepticism, the use of checkmate California red scale pheromones is now the industry standard for management and is keeping and has been keeping California red scale well below economic levels for 3 years running. Thank you.
Thank you so much, David. This is fascinating. So cool to learn about the pheromones and how prevalent and effective they are. Here are the poll questions that we'd like you
Okay.
to, as participants, to answer. We'll give those just a minute for people to read. And if you have any questions for David, please feel free to put those in the chat. Ooh, Devin has a quick question about how does pheromone cost compare to aphytis release?
For the aphytis releases, yeah, Devin, they are because we've gone to the every other row. We are now. It's still like more expensive than aphytis. The thing that what we're hoping is that the use of the pheromone now is cheaper than an insecticide application. And what we're hoping is that growers will go from spraying 3 times in a season to maybe spraying every 3 years. And if the pheromone continues, if the use of that pheromone continues, then the aphytis can come in, and we might be able to see pulling off of the pheromones for another year or two, and then having the aphitis maintain. The disruptors, of course, are the quarantine applications for ACP if you're in those areas. But aphitis is still much cheaper.
Awesome. Okay, we're going to share the results. And David, if you could go over those.
Yeah, okay. So it looks like we've got most everybody recognizing that of the four reasons to pursue looking at pheromones, uh, and it's the factors that I mentioned, insecticide resistance is the most important one. If you take away the ability to use an insecticide, then you've got that sort of in your back pocket management tactic that allows you to clean up a population or meet some sort of quarantine regulation, or, if things just go sideways, you need to be able to have that tool in your back pocket. And if it's not there, because of resistance, then that becomes problematic. And so having the pheromone allows us to use the pheromone, and we know that insecticide resistance is going to go away if you stop using that particular active ingredient, that mode of action. So it's going to be a win-win, but resistance is the key. And then the pheromone helping parasitism. And most everybody got this right too. It's that the female, the third instar female that's before they get mated, is what the parasitoid wasps really prefer. And if you can keep them in that stage, then that is going to help parasitism.
Now I got one more question for you about if it's in the first year of use, is it better to apply to every row and then start alternating the second year?
Yeah, that's going to have to come from the site by site issue. What we know is that if you can get the red scale populations down, if you can start in on your pheromone approach with the populations low, it's going to be effective, the most effective. So it's a matter of getting them down. And if you want to go in with the full complement of pheromones just to get full saturation, yes, because the saturation issue also is impacted by the hedging. And if you're not pruning and hedging and opening up that canopy, then yes, you do need to maintain every tree the 180 dispensers per acre.
Cool. Do you see the potential for CRS to the pheromone to evolve as a means of resistance?
So that if we, what we would do is then look at the lepidopteran systems, like with codling moth, and we don't see any resistance developing there. And that's probably going to be the same thing. It's going to be probably infinitesimally small, the chances of a female having a mutation that changes her pheromone and a male then having a complementary mutation to be attracted to that new pheromone. That's just so unlikely. Irrespective, if the pheromone in these population changes, then all we have to do is get that new pheromone and start synthesizing it and use that new one. So we can always stay up with any resistance that does tend to develop.
Very cool. Does the aphiditis use the pheromone to find the instar 3 females?
That's a great question, Devin. I'm not sure exactly what all uh, the aphids use in their searching. I know that there is cuticular hydrocarbons, there is host plant volatiles, and the third instar females are not the only stage or gender they go after. The Aphidas wasps will also very easily parasitize male aphids. And so there's probably a wide range of chemical cues. But once they have found a colony, they will use tactile responses to antenate and drum around. And when they find a large individual, what they do is they get on top of it and they will circle around, and that helps them gauge the size of the individual. So there's going to be a variety of different cues, both chemical and tactile, that helps them find that stage that they prefer.
Cool. And if populations are high, how much good do the pheromones do? Do they still work?
Devin, if you look at those publications, you'll see that in some of our early experimentation, we had some pretty high, pretty high blocks there, and we got great reduction still, like 60, 50, 60, 70% reduction. So you're getting well on your way.
Excellent. Okay, well, we're going to turn it over to Devin next.
My thing here. Ants and carpenter moths change the canopy and underbark natural enemy kills of mealybugs in grapes. This information is based on my 30-year career as a pest control advisor with thousands of hours observing mealybugs and their biocontrol in vineyards. At the bottom of this slide is a website where I have documented what I learned in various crops. I will show this again at the end and post it in comments afterwards. Next. Vine mealybug has been California's most difficult vineyard pest since it first showed up in the early 1990s. Both conventional and organic growers apply pesticides multiple times and damage still occurs. Yet, under the right circumstances, natural enemies can control VMB without pesticides. The key is ant control. Great mealybugs can also cause problems, but today I am focusing on the vine mealybug. Most of my examples apply to the San Joaquin Valley where I work. Factors that influence mealybug biocontrol include pesticides, climate, geography, and variety and condition of the vines. I speak today on interference from other arthropods, namely ants and carpenter worms. Next. Go ahead. I will illustrate the importance of monitoring and understanding natural enemies and other organisms that affect them. Biocontrol is more than just tossing out predators and parasites from insectaries. Success depends on understanding the system. In the mealybug system, refugia, created by ants or carpenter worms, change the kinds of natural enemies attacking vine mealybugs. A guild is a group of organisms with similar food and location. In this case, the guilds consist of predators and parasites of vine mealybug. One guild works under the bark starting in the spring. A second guild works in the canopy in the summer. Some of these natural enemies belong to both guilds. Next. Vine mealybug overwinters on the trunk and roots, spreads upward for a couple of generations, then moves onto the canopy in a wave of crawlers from late May to mid-July. All stages are always present, although some stages are more common at different times. Some mealybugs always remain under the bark, even through the summer, making a reservoir protected from most pesticides. In August and September, mealybugs migrate back to the trunk to find overwintering spots. Next. Go ahead. Overwintering sites depend on the type of soil and the structure of the vine and bark, which may be loose or tight. In some soil types, most vine mealybugs overwinter on the lower trunk below soil or on roots, typically in an ant nest. In some areas, they overwinter more on the trunk, often in splits in the cork or around lumps of callus tissue. Carpenter worms provide ideal sites for overwintering and throughout the year. These fat caterpillars dig shallow holes in the cork. The mealybugs can feed on the exposed phloem in the cambium layer. Presumably, the worms like the sugar in the mealybug honeydew. Carpenter worms are found only in vineyards with vine mealybugs. This is an example of symbiosis where both partners benefit from each other. The carpenter worms protect the mealybugs from natural enemies. How they do this is unknown, but we have not seen parasites and predators active in the wormholes, except the snout moth caterpillar, as we shall see. This is a good example of a refugia. The mealybugs remain protected in the holes at times when they should be disappearing due to biocontrol. Next. In late May, a wave of mealybug crawlers moves upward into the canopy. They first appear on leaves or fruit that touch bark. Many can be found moving up the canes where they are most vulnerable to pesticides and biocontrol. Eventually, they reach free-hanging bunches of leaves. Next. A number of predators and parasites help control vine mealybug under the bark. These are the 5 most important. The lady beetles, midges, and snout moth caterpillars start early enough to make a big difference before the end of May. Usually, one predominates in any given vineyard. The lacewings and anagyrus parasites arrive later and have less impact under the bark before June. But in August and later, they are both keys to reducing overwintering populations to near zero, even under the bark. Next. Lacewings and anagyrus are the only effective members of the canopy guild for vine mealybug. They can do an excellent job controlling vine mealybug, provided the underbark guild has reduced the mealybug populations to manageable levels. Unfortunately, both of these are highly susceptible to ant predation and interference. I will talk more about these natural enemies after I cover the ants. Next. I subjectively divide vineyard ants into 4 groups based on how effectively they suppress natural enemies. Ant group A is the worst, with fire ants and Argentine ants. Fire ants tend to be on scattered vines throughout a block. These vines will have the worst VMB infestations with almost no biocontrol under bark or in the canopy. Argentine ants need water. They like vineyards with drip irrigation or come into edges next to irrigated yards or wild riparian areas. These 2 ants are also the trickiest to control. Next. Ants in group B cause serious suppression, but some natural enemies can partially avoid them. Field ants and pavement ants are common examples of a large group. Field ants strongly suppress lacewings and anagyrus parasites, but midge maggots, dusky lady beetles, and snout moths can often avoid them. Next. Ant group C includes several ants that like to tend mealybugs for the honeydew, but don't seem to be very suppressive of natural enemies. Pyramid ants look superficially similar to fire ants. They often prey on other ants, such as fire ants and pavement ants. Crazy ants are often found together with other ants, such as Argentine ants. By themselves, they don't seem to cause a lot of problems. Next. I haven't been able to judge how much suppression is caused by group C ants. These are representative. Thief ants are extremely small and tend to mass around mealybugs or girdle wound sap. As you can see, there are hundreds in this picture. Carpenter ants are not very common. They fight vicious wars with neighboring colonies. Next. Anagyrus is specific to vine mealybug and related species such as citrus mealybug. It does not parasitize grape mealybug. Antivirus is a key to biocontrol, but it starts late, not emerging from overwintering mummies until late May or June. It builds up quickly and is an excellent searcher. By the end of August, most vine mealybugs are parasitized, both in the canopy, in the bunches, and under the bark. The overwintering mealybug population is very low, except where carpenter worms provide a refuge from anagyrus and various predators. Anagyrus is very susceptible to ant interference, not just group A, but also group B. If field ants are abundant, anagyrus will have poor success. Anagyrus is available from several insectaries. Next. Green lacewings are excellent predators capable of controlling vine mealybugs almost by themselves. But ants strongly interfere. Forget dumping on lacewing eggs from insectaries. Ants eat them for breakfast. The egg stalks probably offer some protection, but not enough. Green lacewings show up when enough honeydew accumulates to attract the adults. Dr. Chris Tollerup, in his 2000 dissertation, 2007, in Coachella Valley, showed that when field ants are controlled, vine mealybugs almost disappear. I have seen similar cases where, in the absence of ants and carpenter worms, lacewings are capable of taking down heavy infestations of VMB quickly. I know one grower who has not applied any pesticide sprays for mealybugs in many years in both organic and conventional vineyards. Next. Dusky lady beetles are a large group of Coccinellidae in the tribe Scimnini. Many of them are excellent mealybug predators in the underbark guild. The species varies with geography. Cryptolaemus montruseri, the famous mealybug destroyer, is prevalent along the California coast. You can buy them from insectaries. Nephis sordidus, the little brown mealybug destroyer, is better adapted to the hotter summers and colder winters in the San Joaquin Valley. It is also not hard to rear, but it is currently not for sale from insectaries. One or more species of Scymnus is also found in the Central Valley, especially between Sacramento and the coast. Next slide. Nephis and the other dusky lady beetles are moderately tolerant of ants. Fire ants will decimate them, but they can survive on vines with field ants and provide reasonably good control. It helps that they hide under the bark. The larvae have tufts of wax that make them resemble mealybugs. I once saw a field ant grab a Nephis larvae and come away with only a mouthful of wax. The larvae can be found among the mealybugs. They show up as early as late April, and mealybugs often become scarce 3 or 4 weeks later. Adults move quickly and can be mistaken for other small brown beetles. The dark triangle at the front of the carapace is distinguishing. The pupa here is upside down. Old pupal skins indicate populations are present. Nephis does not control mealybugs hunkered down in holes of carpenter worms. Next. Midge maggots are the most important predator of grape mealybugs. For some reason, they are not as common feeding on vine mealybugs, but when they are present, they can quickly clean up an infestation under the bark. As shown in the middle picture, you can see the arrow points to a midge maggot underneath that pile. To me, they seem to do better when both mealybugs are present. The larger maggots, pupae, and adults can be spotted by their bright red color. The small maggots are near white. You can see a small maggot on the mealybug in the upper left there. The arrow points to it, okay? And then it'll just simply consume that entirely mealybug as it grows. Uh, All maggot sizes attack all sizes of mealybugs. Eggs can be laid on or near mealybug egg masses or directly on large crawlers. First generation maggots appear under the bark as early as March, but are more common in May and June. Their presence predicts good mealybug control. Midge maggots are moderately tolerant of field ants, at least partly because they are hidden. They do a good job of cleaning up the grape mealybugs found under grapes that are touching bark, where mealybugs are protected from other predators and from pesticides. Next slide. In blocks with a lot of carpenter worms, the only effective underbark natural enemy is the snout moth caterpillar, an undescribed species of Alphaeus. They are typically found nearby or sometimes inside the wormholes. In the picture of that has the carpenter moth, you can see the medium-sized uh, Alphaeus up to the right in the shadow there. The caterpillars resemble leaf rollers. They can be distinguished by the two dark blotches on the pronotum. You see those? Or sometimes it's a solid line all the way across. Snout moths are moderately tolerant of field ants, but not fire ants. Next. I have a web page. It's at the bottom, and will also be in the chat, and where I document what I've learned in my years. And there's three papers on there that would interest you for further information. One is about the predaceous gall midges, the second about the little brown mealybug destroyer, and the third about the mealybug feeding snout moth. Next. And I also have information. Oh, I'm sorry, go back. I have information about ants there, including one that talks about the biology of ants in the San Joaquin Valley and vineyards, and another one about mixing ant baits. And in the chat, there is, or there should be, did you put it in all in here yet? Okay. So I've written a more detailed account of what I just talked about. It's not quite finished yet, so I haven't posted it, but I'll post it very soon. It's called Ants, Carpenter Worms, and the Canopy and Underbark Guilds of Mealybug Natural Enemies in Grapes, and that is in the chat if you want to, and will also be on my webpage soon. Okay, that's
Awesome.
about it.
Thank you so much, Devin.
Sure.
We'll get the polls up next. And one question that I have is like, there anything that we can do to attract these natural enemies?
Some of them are for sale, like I said, Anagyrus, the lacewings, and controlling the ants, of course, which is not easy, but it can be done. And I would advise, for example, some organic growers have taken to peeling the bark every year and then spraying with oil. To me, that's not a good idea because many of the biocontrol depends on those underbark predators. So if you peel the bark, they're not going to be as effective. I can't think of anything else right now.
Okay. Can end the poll here and have you go over the results.
Pardon? I'm sorry. I only see the question, will anagyrus feed on host honeydew?
Oh,
And I I'm pretty sure that they do. I don't know why they wouldn't, but I don't specifically know the correct answer to that. But I would be surprised if they didn't.
and then the poll question, a group of animals with similar food and habitat is a guild, correct?
Uh, yes, that is correct.
And then the mealybugs are protected from biocontrol by ants and carpenter worms.
That is also correct.
Perfect. Sounds good. And if there are any other questions, please put them in the chat. Is there anything that, like any sort of habitat that can be added to farms that would attract any of those natural predators, like the mealybug destroyer or anything like that?
Uh, like I said, they like bark, and I will mention that uh, grape varieties differ in how loose their bark is. And I have never done exactly experiment on it, but my my I strongly suspect that the biocontrol works better on vines that have loose bark rather than the vines that have very tart bark, like Thompson seedless, that are it's really hard to peel back. Uh, and also, young vines do not have very much bark, and so the biocontrol has the underbark guild has a hard time. And also the uh, vine mealybugs, they also like to hide under the bark. Uh, so they may move up more quickly than they would on a mature vine. Of course, the small vines also, it might be easier for them to reach the phloem from the cracks in the bark. And I've never done it, but I've kept trying to persuade growers to wrap something around the small vines, like a piece of burlap or something, so that the mealybugs would come up there and stop, and the biocontrol agents could get under there and do their job and still be protected somewhat from pesticides and the ants. But that's just a suggestion. I've never got anybody to actually try it.
Excellent. Joanne has a question about, have you seen the biocontrol working better with cover crops?
Um, that's a good question. Um, I would say some of the natural enemies, particularly lacewings, move back and forth between the cover crop uh, and the vine. So in that case, they might help. Uh, and I suppose trying to, I don't. Other than that, I really don't know. So, whether any of the adults, for example, the larval stages usually do the most feeding, and they don't move back and forth. But perhaps maybe the lady beetles or parasite adults might visit the cover crop. But I think they're mostly interested in the vine, where they're looking for honeydew. They're attracted to honeydew. There are some generalist predators that move up from the detritus or the cover crop. For example, um, earwigs, they might move back and forth, and they help some. They're not very important, but they might help some. I think the what the cover crop does. This is what Kent Dane found with leafhoppers: is reduce the temperature in the vines, and that might help with biocontrol. Uh, make the giving a better habitat for the predators and parasites. That made more of a difference um, than actual moving back and forth. Okay, Mr. Gross. It's interesting. Yeah, about the codling moth. Uh, that's true. And that's a good way, but that's a little different than this case. You would want to leave the cardboard or the burlap on the vine so that the biocontrol could continue to work. It's I don't I don't see it as a control measure to take it off and smash all the mealybugs. That would be an enormous amount of labor, and I don't think you'd get them all anyway. I think they would continue to move up. But interesting idea.
Well, thank you to David and Devin for sharing your knowledge. Thanks for everyone for attending. We'd love to have your feedback. And if you need those CE credits, we need your license numbers. And the DPR requires you to take a quick 10-question quiz. There are additional resources on the website that are recommended by our speakers, including an article on the future of organic insect pest management. And I don't see any other questions in the chat. Thank you both so much.